Hello, everybody, and welcome back to BioSC 140 Human Physiology. This is the video for Laboratory 7/8, Osmosis and Diffusion. This is the first video that's going to be over the experimental procedures and kind of some background information. So I want you to take note of this first screen. In some experiments, some laboratories in this class, there's just one procedure that we do per class. This one has a whole bunch. So you need to understand that and compartmentalize each experiment. Study them, study Fox 2.6a and then put that away in your brain. Study 2.6c and put that away in your brain so you don't get them mixed up. Remember, I want you to be able to tell the general story of all of the labs that we do, like the procedures that we do, on top of knowing all of the, the science behind it all. So with that being said, let's jump right into Fox 2.6a. So in Fox 2.6a, we're going to start out by grabbing a test tube and putting 2 milliliters of H2O in. So H2O is water, and you should know a few things about its shape, about its polarity, about its chemical makeup. We're also going to add 2 milliliters of this molecule over here called xylene. It's called xylene. I want you to look at the structure of xylene right now. And I want you to, it says it right below. I want you to think about the polarity of that molecule. And I want you to think about what's going to happen when we mix these two or put both of these into the same test tube. What's going to happen? Are they going to mix together well? All right, we'll get into that. Actually, let's just kind of jump into it. So xylene is nonpolar. Water is polar. Do polar and nonpolar substances mix? No, it's like oil and water. It's like vinegar and oil for a vinaigrette salad dressing. Polar and nonpolar molecules do not mix well. And what we're going to see is we're going to see two distinct layers. Now, xylene is a clear liquid. You're probably fairly familiar with glasses of water. Water is also a clear liquid. And just by looking at the two layers, when we do it in lab, like you can tell there's two layers, you can see the interface between the two, but you can't really, there's no color difference and there's no viscosity difference. There's no way to really tell which layer is water and which layer is xylene. You just see clear liquid on top of clear liquid. So what could we do to figure out which layer is water and which layer is xylene? Well, let's move on to step 2. Step 2 is to add a dash of potassium permanganate, KMnO4. So KMnO4 is a salt, and it's a bright purple salt. It's very colorful. And when you put it in water, it dissociates into potassium and permanganate, hence the name. What is this bright purple salt going to do? Well, this salt is going to go into the water layer and it's going to dissociate. It's going to dissociate. And when it does so, it's going to become charged. It's going to become 2 ions. Ions mix well with polar molecules. So they're going to hang out in the water layer and they're going to avoid the xylene layer. And they're bright purple. So now our water layer is bright purple. So we know which layer water is. We've got the clear xylene layer and we've got the bright purple water layer with potassium permanganate dissolved in it. Next up, we're going to add a milliliter of vegetable oil. So vegetable oil is a lipid. It's an unsaturated triglyceride. What is vegetable oil going to do? What is vegetable oil going to do? Well, does oil and water mix well? No. The oil is going to interact with the nonpolar xylene. Nonpolar things mix with nonpolar things. So vegetable oil has like a yellowish color to it. So now our xylene layer has a yellowish color to it, and our water layer is like bright purple. Next up, so we've got these two layers. We've got the non-polar yellowish layer. We've got the polar bright purple layer. Next up, we're going to mix them. We're going to put a cover over the top of the test tube, and we're going to shake it. We're going to shake it a whole bunch so that it all mixes together. And what are we going to see? What do you think is going to happen when we shake this test tube a whole bunch of times? Well, it's going to look kind of like this test tube right here, where you see little circles of purple and little circles of yellow and areas of purple and areas of yellow. It's going to be all jumbled up. But very, very quickly, the two layers reform. Like really quickly. Like we see it happen before our eyes in a matter of a couple seconds in lab. Polar mixes with polar, nonpolar mixes with nonpolar. Next up, we're going to add a soap. So I told you we're going to talk a lot about soaps and amphipathic molecules this semester. So what does amphipathic mean? It's come up a few times. It's underlined and bolded. It is a vocab word. An amphipathic molecule has a significant polar region and a significant nonpolar region. Soaps have amphipathic molecules. Well, what the soap is going to do, or let's get to that later. So we're going to add this to our test tube, and we're going to shake it again. So we've got our nonpolar layer that's yellow, our polar layer that's purple. We're going to add a soap, and we're going to mix it again. What's going to happen after we've added soap? Well, the layers are not going to separate out again. They're going to blend together. We're going to have one solution that's a blend of the water layer and the xylene layer. It's all going to blend together. And why is it going to blend together? We'll talk about that in the discussion. Think about it though. All right, so moving on to FOX 2.6c. So in this lab, what we're going to do is we're going to get some dialysis tubing. So dialysis tubing are these clear little packets, clear little tubes. And the walls of these tubes, they look like this. They look like this. The walls of these tubes allow water to move through, but nothing else. So salt cannot move through these tubes, through the walls of these tubes. Glucose cannot move through the walls of these tubes, but water can. We're going to close off one end of the tube. We're actually going to get a bunch of these tubes, and we're going to close off one end with these clips. And then we're going to add 12 to 15 milliliters of water into, or of a solution into these bags. And then we're going to close the other end with another clip. So we're going to have a clip, 12 to 15 milliliters of a solution, and then another clip. We want to make sure that there's still some room for expansion in the bag, so that there's still some like area for the bag to grow. We want it to be kind of floppy, leave room for expansion. So we're going to have 5 bags. One bag is going to have deionized water only in it. One bag is going to have a 10% sucrose solution. One bag is going to have 20% sucrose solution. One bag is going to have a 30% sucrose solution. And one bag, again, is going to have 0% sucrose, so DI water. So 2 DI water bags. And then 10, 20, and 30% sucrose solution bags. We are then going to prep beakers. and we're going to have 5 beakers total, 4 beakers with DI water, 1 beaker with 30% sucrose. And we are going to submerge our prepared dialysis tubing bags in those beakers. So this is the setup. We're going to have 1 bag with distilled water and distilled water in the beaker, 1 bag with 10% sucrose in distilled water, 1 bag 20% sucrose in distilled water, 1 bag 30% sucrose in distilled water, 1 tube with distilled water and the beaker having 30% sucrose solution. So this lab is entitled Osmosis. What do you think is going to happen? You should know a few things about osmosis based on the lecture by this point. All right, so we're going to measure the weights of the bags at time 0 using a scale, and then every 15 minutes, we're going to pull the bags out of the solution, measure their weights for about an hour and 15, hour and 30 minutes. What do you think is going to happen? All right, next up, this is our first time using blood samples in this class. Now, we're online, so we're not actually going to be doing this, but if we are in person, this would be the first time that we retrieve blood samples from each other. No, it wouldn't be. This would be the second time. I'm sorry about that. Yeah, so this should be old news. And use universal precautions when obtaining blood samples in lab. Assume every bit of blood is hazardous. All right, so this is going to be Fox 2.6C S2. We're going to get 9 test tubes. We're going to get 9 test tubes. We're going to wash and disinfect them with bleach before we're using them. Make sure they're clean. Make sure they're clean. 1 dropper full, about 2 milliliters of solution. So test tube 1 is going to have pure water. Test tube 2 is going to have 0.41 grams per deciliter sodium. Test tube 3 is going to have 0.82 grams per deciliter sodium. These solutions are going to be pre-made in lab. All we have to do is add 1 dropper, about 2 milliliters of the solution in the corresponding tube. So 9 tubes, these sodium solutions in 1 through 5, these glucose solutions in 6 through 9. Then what we're going to do is we're going to add a drop of blood to test tube 1. Mix, make a slide with that solution, that blood and water solution, and we're going to look at what the red blood cells look like. We're then going to put a drop of blood in test tube 2. Mix, make a slide with the solution, and look at what the red blood cells look like underneath a microscope. Test tube 3, same thing. Drop of blood, mix, put underneath a microscope, and see what the red blood cells look like. We're going to do that for all 9 test tubes. What do you think the red blood cells are going to look like? Do you think they're all going to look like normal red blood cells? What's going to happen to them? How will they look? We'll talk about that in discussion. Oh, nope, we'll talk about it right here. Some cells are going to shrink, they're going to crenate, they're going to shrivel. Some cells are going to be normal shaped because they're in an isotonic solution. Some cells are going to be round, like ball-like, and some might actually even break apart or lysed. What is going to determine what's the fate of the red blood cells? Can you predict what solutions will have red blood cells that look like each of these? Remember, think osmosis, or think tonicity and osmosis for this one. All right. Next up, we have Fox 2.6C S3 plus 4. So we're going to be looking at membrane permeability in this one. So we're going to have 8 test tubes total, washed, cleaned beforehand. The first 4 test tubes are going to have urea, ethylene glycol, glycerol, and glucose. The next 4 test tubes are going to have methyl alcohol, propyl alcohol, ethylene glycol, and glycerol. We're going to put 2 milliliters of each solution in the test tube. And just like the last lab, we're going to add a drop of blood to test tube 1 and mix. Just after this time, after we do this, we're not going to look at it underneath a microscope. After we put the drop of blood in the solution is going to be kind of dark. We're not going to be able to see through it. And what we're going to do is it's going to be dark. We're not going to be able to see through like this. What we're going to do is we're going to hold that test tube up against the lab book, and we're going to start the timer the moment we mix the blood and solution. We're going to start the timer when we do the mixing. We're going to stop the timer when we can see through the sample. So right here, we cannot see through the sample because we cannot see the letters behind it of the lab book. Right here, we can see the letters through the test tube, so we're going to stop the timer. When red blood cells are whole, when they're their proper shape, they're going to obscure the light that travels through the test tube, and it's going to make it so that you cannot see through the test tube. These solutions, we'll get into the details, but these solutions are going to lyse the red blood cells. They're going to break the red blood cells. When the red blood cells break apart, when they lyse, they're not going to obscure the light so much that passes through the sample. So we're going to be able to see through the sample and see letters through the sample. So you stop the timer when you're able to see the letters through. We're going to record the time it takes from mixing to the solution being clear. We're going to call that the hemolysis time. We're going to call that the hemolysis time. So can you predict the hemolysis time for each of these samples? What do you think is going to affect the hemolysis time of these samples? We'll get into that with discussion. So I'll give you a little more insight before we go to a discussion. What we're looking at with this first group is we're really trying to look at molecular size. Now, molecular size is really hard to calculate. Molecular weight is much easier. So we're using molecular weight as kind of a proxy, an approximation for molecular size. So does size have an effect on hemolysis time? With the second half of the test tubes, 5 through 8, we're going to be looking at the presence of polar groups. Now, there are size variations, but there's also significant polar group variations. Does the presence of varying number of polar groups, polar functional groups, does that affect hemolysis time? All right, lab notes. Perfect. I will see you in the next video, which will be the discussion video. Email me if you have any questions, everybody.